So we've been doing a lot of stuff remotely. I haven't heard from a lot of people. I haven't checked in with a lot of people. So what have we missed? What do we need to catch up on? Cool. Like, you tell me. So we covered the topics, kind of an interesting set of topics for us to have done remotely. I always feel a little weird doing central dogma in genetics because I know it's a topic that gets covered in other classes, and it obviously is central dogma. This is a topic that you'll probably encounter in a lot of different contexts. So when I taught biology 141, the introductory biology, I would teach this content. I know it gets taught in cell biology, and I'm sure it shows up in biology 142. So just for this class, just another reminder, we cover this from a genetic standpoint. So when I talk about that, I'm talking about like meaningful differences that can be inherited. I feel like that's more at the core of genetics. If you get into, I guess other people who might teach this might focus a little more on the molecules. I focus less on them because I know they do come up in other classes, and it's kind of a topic that I think some people quite enjoy and some people really don't. And I don't want to keep hammering this molecular biology concept at you in all kinds of different classes if you're not, I don't know. Like if you had it in introductory biology and you didn't like it, you didn't get it, and I have it for you again, we could spend a lot of time on this, and so we just kind of go faster through it. So it kind of bridges into the field of what's more properly called molecular biology. So we talked about molecular genetics, so emphasis on the molecules like this one. I think thinking about molecules is a learned skill. So if you take a biochemistry class or we have an upper level molecular biology class, these are classes where you think about the molecules and how they interact. And maybe it starts making a little more sense the more times you cover it. For us, kind of want to just revisit the steps, and we can think about them starting today kind of in terms of how things might be affected if you mutated these. And that's a little more in the realm of genetics, I think. So I'll give you my mostly brief review of central dogma. We do have, I don't think I really looked at the assignment that was due last week. That was a self-graded one. So if you had issues about that one, I saw a lot of 100% on that one. So good job there. There's an assignment due Thursday of this week. So if you did have questions on that, today would be a good day to ask them. And then a week from Thursday will be our next exam. What are we at? Exam 3 now? So I don't have the content on Canvas yet for you to review, but I hope to get that up soon. I've been a little short on time. I hope to make some ground back on this. All right, no questions? Did you do genetics light over the time when we weren't seeing each other? Just the minimum? All right, so there's a kind of an overview of central dogma that I like to do. We could do that now. So here we go.
Professor?
Yes.
Can you share your screen?
I would love to. Thank you. I knew that I would mess up something on technology, and there we go. How's that? Is that working?
Yeah.
Cool. Well, we got tired of just watching me up here. All right, so central dogma, what are the molecules involved? Big categories of molecules. You should long to use your vocal cords these days. Jenna. DNA. Right. DNA becomes mRNA, or we'll just go simpler with RNA. And in typical fashion, we typically think about this ending up in protein. So I guess back to genetics, I think I mentioned this once before. You could think of the DNA as your genes. These are the instructions that your cells were given. And protein end up giving you functions. And phenotype. So we don't inherit proteins from our parents, but we need the proteins to do things. So if you have an enzyme that's, say you have an enzyme that breaks down lactose for you, you get that from your parents, you get the instructions for making that enzyme. And if you get a different version of the lactose digesting enzyme instructions, this is the basis for being lactose intolerant. Actually, more of a natural condition for people not to digest lactose, more of an ancestral version. So a lot of times I refer back to the fruit fly labs. So having a functional pigment transport molecule, you have the instructions for making that pigment transport molecule. You inherit those from your parents, your daddy and mommy fruit fly. And then those instructions allow you to build the functional pigment transporter or whatever you're making. So that ends up being the phenotype. So if we have a fly that can't make the red pigment, that's because it doesn't have the red pigment making enzyme, and that would mean that it has a faulty version of the gene for that enzyme. So that's really why we're interested in central dogma from a genetics standpoint. So we're all still basically grounded in phenotype, but we can't cover genetics without talking about molecules, especially these days when molecules are all over the place. DNA is really central to people's investigations in a lot of fields of biology these days. Okay, so the central dogma has the molecules, has the processes of converting them. So what's the process of converting DNA into RNA? What's that called? Transcription. And RNA to protein. Anybody else want to talk today? Translation. All right, so transcription, translation, kind of like mitosis, meiosis, these words are deceptively similar as you write them. Transcription, the script part of that is like writing, so it's sort of like copying somebody's notes. So going from DNA to RNA, that's sort of like copying the same notes into a different handwriting, right? So it's still letters of nucleic acids. The DNA nucleotides become the RNA nucleotides, and it's one for one, really. So if you have a DNA sequence that's really messy, 5' GC A T, then in the RNA that gets copied as it is, assuming we've got the strands correct here, GC AAU dot dot dot. So that strand of DNA that is the, we call the coding strand, that becomes the same sequence in the same direction of nucleotides that show up in the RNA molecule. But then this translation, I don't know, kind of nice words if you think about how we use these in the English language, right? So transcription, to transcribe something is really to take words in the same language and put them into a different format. But translation is to take words from one language and make them into another language. And they don't look the same anymore, right? So the nucleotides of RNA don't look like what are the building blocks of a protein? amino acids. Everybody awake this morning? Cool. Right. So our translation of this mRNA molecule is going to look very different from the sequence of nucleotides that were the instructions for it. So this process of converting from RNA, or mRNA in this case, into protein, how does it work? Well, we take this molecule, 3 letters at a time, and these 3 letters become a codon for one amino acid. Probably not something I can really draw for you right here. All right, questions on any of that? I do have the poll anywhere up if anybody wants to weigh in on this. What do you say the humidity is in here today? Those of you at home, it's a little rainforesty here today. Right? Not just me? Cool. It's November. They have a plan for heating the building, not cooling it down. All right. So this is the overview of central dogma. It should be very familiar to you. We can look at the molecules. We sort of zoom out on these molecules. Let's say we start with a double-stranded DNA molecule. So now not looking at the individual nucleotides, but this DNA would be made up of lots of nucleotides and usually thousands of nucleotides. So more than I can draw for you here, but just sort of imagine we're doing bigger picture. So what does it take to get that DNA transcribed into RNA? What are the signals involved on the DNA molecule that cause this transcription process to happen? How does your cell know where a gene is? Anybody at home want to turn on their microphone?
It was the promoter region that helps it was RNA polymerase, right? That starts the whole thing, sort of.
Okay. Yeah, so that was a couple of good vocab words that are both involved. Promoter region is a term for a sequence of nucleotides. I don't know how I want to do this. All right, I'm going to draw a box around it. So some part of this DNA molecule is a sequence that is not a sequence that contains instructions for amino acids, but it's a sequence that has a certain sequence of nucleotides that are recognized by enzymes that start the process of translation. So the promoter is on the DNA molecule, and for the most part, the promoter does not actually itself become transcribed. And the promoter is responsible for recruiting the enzyme there called RNA polymerase. And the RNA polymerase starts copying DNA into RNA. So from some start position determined by the specific locations of nucleotides in the promoter, starting at that start position, that's where the RNA polymerase begins to transcribe. Now I said start, and I try to avoid saying start when we talk about transcription. Why is that? Because this step of the process does not involve a start codon. This is one of the things that I have to enforce a few times. I think there's a tendency, oh, the process starts, must be start codon. As we'll see, the start codon is not actually involved here. RNA polymerase doesn't even know what a start codon looks like. Doesn't care. So we'll see the start codon in a little bit, but not immediately. So we had a signal to get the transcription process started. That was in the promoter. What term do we use for the signals that cause transcription to stop? Not a stop codon. Promoter 2. I made like 9 movies about it. The Terminator. So we didn't cover the Terminator in much detail, and I really don't emphasize it much. It just isn't. I don't know, isn't it quite as clean to teach about? So some combination of factors cause the RNA molecule to stop being made at this region that we call the terminator. So between these promoter and terminator regions or sequences, those determine where the edges of this RNA molecule is made. And we can do this in a way that's probably most familiar. We're going to do this in eukaryotes. So after transcription, we end up with a first draft of our RNA molecule. And the molecule that's produced by transcription often gets called the transcript. Then what? Straight to translation or something else? Take out the introns. All right. Yes. So after this process, there is a shortening of the molecule. So I drew little hashes in there like those were nucleotides, but really this scale is much bigger than that. So we kind of diagram in parts of this. And let's say we've got just a couple of introns. So parts of the initial RNA molecule will stick around. And those are called the exons. And parts of this will get removed. Those are called the introns. How does the cell know how to take out an intron? Or where? So if you remember, the introns had their own specific sequences. Maybe most memorably, there are sequences just inside of the ends of this intron that are also interpreted by enzymes that say, this is an intron. This needs to be removed. So we saw this before. I think this stuff is fascinating, personally. I think when you first encounter this, it's sort of like, yeah, the cell knows what to do. These little enzymes, they don't have brains. They're not little organisms. They're just molecules, and they can only work with molecular interactions. So the enzyme that takes out an intron has to know where to go, but it doesn't know anything, so it has to match up with its target. So it matches these short nucleotide sequences that are distinctive as intron removal sites, splice signals. So they are present there in the intron, and they say, take me out. Take me out for a nice dinner as soon as this pandemic is over. Okay, so if you can't tell, I'm trying to focus on the signals here. You should have had the process before you know about splicing, but let's look more deeply about what's happening to cause the splicing. Okay, so introns cut out. There's a little bit of more modification. Some people insist that I include this information. So there's a little bit of modification in eukaryotes. On the 5' end, there's a methylated cap. On the 3' end, there's a poly A tail. These help in signaling to the cell that this is a molecule now ready for translation. So when it's ready to translate, then I'm going to give it the official title of mRNA, messenger RNA. Sometimes I call it the mature mRNA. It's had some time to grow up. All right, so that molecule there is ready for translation. What molecules are involved in translation? Oh,
tRNA.
tRNAs are involved. All right, I'm going to draw a little schematic tRNA molecule. Yes, tRNAs are involved. So we've got mRNA, tRNA. tRNA is responsible for bringing in the amino acid and matching a codon somewhere on that molecule. What else? How does the tRNA know where to go? The ribosome. Okay, so the signals for translation also cause this ribosome, big old molecule, to clamp onto the mRNA. And the ribosome's job is to match up the tRNA with the mRNA in specific locations. So the ribosome is going to go down the mRNA, kind of zips down this molecule, and as it goes, every 3 nucleotides, it matches up a new tRNA with the corresponding codon. Right, so the ribosome goes down the mRNA molecule. tRNAs bring in new amino acids. Those amino acids get joined together in this purple polypeptide chain. And from start codon to stop codon, that determines the length of the polypeptide chain. So I've kind of run out of space here. But on this tiny molecule, somewhere in there would be a start codon, somewhere in there would be a stop codon. And the codons get read starting with the start codon. The start codon is not the first 3 nucleotides. So on this mRNA molecule, there would be other nucleotides over here that are not translated. Sometimes they call that the untranslated region. I don't know why. Because it's not translated. All right, so start codon, then the next 3. So AUG is our standard start codon. Next codon is CCC, then ACG. And then continuing down this until we reach a stop codon. All right, I'm going to try to do more of a schematic here that just emphasizes the signals involved. See how I can do. All right, so maybe we'll work from the bottom up. This is our mature mRNA. So starting at the start codon and ending at the stop codon, those become the instructions for putting amino acids together for the protein. Oops, I don't want to do that. Delete. I'm going to do it over here. So the signals that are read in the mRNA for start and stop of translation, those were present already in the previous RNA molecule, along with an intron. I'll just leave it at 1 intron. Go a box in there. So on the RNA molecule were the signals to do the splicing. And everything that is a nucleotide in the RNA was contained in the instructions of the DNA. And then the DNA would have the extra signals of promoter, terminator, and if you look at the DNA sequence, all these signals are still present here. So they're in the instructions, but they're not used while this is a DNA molecule. So I don't know, this helps me to think about the whole process, and I think if you understand all of this in detail, that really means you can explain the process to somebody. So sometimes I ask the question, possibly on upcoming exams, what are the signals? In what molecule do you find them? And sort of where are they recognized? So in the DNA, if you look at the DNA molecule, now that we've reconstructed that, all the signals are present there. We have promoter signal, we've got start codon signal, we've got splice signals, stop codon signal, terminator signal. Only the promoter and the terminator are interpreted, are read by molecules while this is a DNA molecule. So the promoter causes RNA polymerase to get involved, copies this into RNA, and it copies the start codon, it copies the splice signals, it copies the stop codon into this first RNA. The promoter, for the most part, is not present on that RNA molecule. The terminator, for the most part, not present on that RNA molecule, and certainly not interpreted on that RNA molecule. So they did their job. So moving down, going from initial RNA to mRNA, still have the start codon, stop codon. They're not read yet. So start codon, stop codon are not ready to be read by the molecules yet. In a eukaryotic cell, until this is a mature mRNA, the ribosome doesn't treat it like one. So the start codon, stop codon, they're still there, they're just not, they're not giving their marching orders to any enzymes right now. But the splice signals are involved. Your book talks about some, I don't really dwell on it much, but there's some signals also toward the 3' end that are involved in causing that poly A tail to get attached. So here in the initial RNA, the splice signals are interpreted, causes splicing to happen, the intron is removed, and then finally we get this mRNA molecule and the start codon, stop codon that have been present in the nucleotide sequence the whole time. They finally get to do their job and recruit tRNAs to make this protein. I don't know, what do you think of that? Amazing! Whew, time flies. Quiet day in the old genetics class. I mentioned eukaryotes. I think we can go over bacteria also. Be a little bit quicker. That's not pretty. All right, so bacteria also have genes in their DNA. They also have promoters. But as bacteria typically do things simpler, their RNA is pretty well ready to go after transcription. If you remember, bacteria had a different signal to recruit the ribosome. You remember what that was called? So in eukaryotes, the ribosome is recruited by the 5' cap and the poly A tail. Those don't exist in bacteria. So instead, bacteria have a stripped down version. They have this SD sequence. So it's another one of those distinctive nucleotide sequences that's involved in recruiting the ribosome. So kind of handy. It's got a rather simple designation there. As we saw in bacteria, it's possible for a bacterial RNA to contain instructions for more than one protein. So let's just do an example with that. Let's say we have this mRNA. It might contain the instructions for making one gene here, one protein, I should say. Run a little bit out of space here. So an important bacterial difference, sometimes the RNA contains instructions for more than one just independent protein with its own SD sequence, with its own start codon and stop codon. When we talk about gene expression, these will come up again as an interesting bacterial feature. So I'll just leave it at 2 there. 2 proteins made by 2 sequences that happen to be encoded on the same RNA molecule. All right, somebody's got a question. Anybody typing in the chat box that I can't see? Whew, I thought it was quiet before. Okay, I'm going to assume you are all experts at this. All right, I'll give you a chance to weigh in anyway. Dun, dun, activities. We're going to do some polling. Polling, 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 polling. Okay, so don't answer these, but real quick, I have a mRNA molecule, and this question was meant, well, Maybe we could do it. Why don't I just walk you through this one? This question was meant to have you looking for a start codon. So one temptation is to just jump right in and start translating. We're a little bit handicapped by me not wanting to type and you probably not wanting to read 800 nucleotides. So sometimes I write a shorter sequence here. So this is a shortened version of what you might see in an mRNA. But it would have, if this is a eukaryotic gene, would have its 5' cap. And then somewhere downstream of the 5' cap would be a start codon, AUG. So codons, including the stop codon, AUG, there's 1, UCA, 2, GUC, 3, GCC, 4, UAG, that looks familiar. UAG over here is a stop codon. So in my little imaginary sequence here, I've got 5 codons total, 4 amino acids, and 1 stop codon. So let's say this is the sequence. Now I've got little dots on there to show you where the codons are split. What would be the amino acid sequence produced by this mRNA?
Do you want us to answer in polling everywhere?
That's what I was going to check. Yes, that's the easiest way for. This is my way to give me a break of talking, give you a break listening to me, give you a chance to interact. Give you a chance to see if you know what you're doing. All right. 6 people going in for A. Let's see how that looks. So everybody says methionine for the first one. AUG, start codon in green here, methionine. Next one. So right after AUG is UCA. UCA, serine. So this isn't meant to be challenging. I'll try not to throw you too many curveballs. This is a sequence as you would read it if you were accustomed to reading nucleotide sequences. 5' on the left, 3' on the right. And that is how the codons are written in the table. So I guess if you want to think about it, this is as much how we choose to think about them as how they really exist in the molecules. So we typically write them as 5' to 3'. So UCA was serine, followed by GUC. GUC valine. So methionine, serine, valine. I've already got one that's the only one with that answer. I could double check the last one here, GCC. Alanine, there it is. So methionine, serine, valine, alanine. So if you're ever entering these, I'll just remind you to use the abbreviations that are given to you. Either sometimes you get a 3-letter abbreviation, sometimes you get a 1-letter abbreviation. There are some amino acids that are deceptively similar. So there are 3 ones that start with GL. There's glycine, glutamates, and glutamine. There's asparagine and aspartate. So if you get the wrong letter in this abbreviation, you could be indicating the wrong amino acid. Okay, a little bit more challenging here. What does an anticodon look like? So here we've got a tRNA that brings in glutamine, GLN. Where is that on there? Here's some glutamine, 2 of them. So the codon is CAA or CAG. Let me, I guess, clear the answers here. Clear responses. And, all right, so glutamine GAA or GAG? Five prime, 3 prime, 5 prime, 3 prime. GAG, okay. So if that's the codon, in the codon table, they're written 5 prime to 3 prime. That's the sequence you would find on the mRNA. What molecule has an anticodon? Oh, there it is. So anticodons are found on tRNA. Codon, mRNA, anticodon, tRNA, and the anticodon is a reverse complementary sequence. So possibility here, what is complementary to these? Right? You're like, I knew that. So complementary, CUU or CU. Am I doing this right? Doesn't look right. All right. No, none of that looks right. Am I doing this right?
I think you wrote GAA instead of CAA.
Oh, thank you. I was copying. See? Perfect example. GLU, glutamate, was the ones I was copying. Should have been copying glutamine. Dag. All right. Thank you for that. Dun 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 dun. This will look better than me crossing everything out. Nope, you don't have to cross everything out anyway. Okay. Everybody at home knew that. Glutamine, CAA, CAG. CAA, CAG. Well, that's a good illustration of test-taking strategy. Get your answer first before you start guessing. My answer was not going to show up. Okay. So these are our glutamine codons. Again, this would be the mRNA. And across from those, GUU, GUC. All right. So now we're getting some GUU. Is what on there? Is that right? Is that the whole picture? That was the pesky 5 prime. We should double check because there's one that says 5 prime GUU. There's one that says 5 prime UUG. Darn, where does that 5 prime end go? Right side, left side. Flip a coin. Does it help if I put arrows on here? So 5 prime to 3 prime. Anybody? All right, so shorthand. anytime nucleotides recognize each other and form a complementary base pair interaction, whether that is DNA to DNA, DNA to RNA, or here, RNA to RNA, every time that happens, it is anti-parallel, so running with the backbone in opposite directions. So this part of the tRNA runs in the opposite direction. That would give it a 5' and a 3', right? That's one of those things people don't like me for. So the way to write this, if you're writing it from the 5' direction, that's 5', then UUG, then 3'. So it's more B than A. Does that make sense? All right, do you remember wobble pairing? So in RNA, it's allowable. It's not as good. It's not good as a proper, I guess I'll put it on here. You don't have to know this term, but I need a term to put here. Canonical base pairing. So that's G pairs with C, A pairs with U. And there was one exception called wobble pairing. What were the two nucleotides involved there? Oh. A and G? A and T. So A and T would be DNA. The wobble pairing is allowable in RNA. Get a visual up for you. Where are you at there? And so the wobble pair is G pairing with U. I guess the wobble indicates that it's a molecular interaction that kind of fits, but not the best. So it kind of jiggles around like, this isn't my favorite place to be. It'll do for now. It's like letting somebody else choose the restaurant, right? All right, it's food. What we saw with wobble pairing was that was one way to get more bang out of your tRNAs. So if a single tRNA can recognize more than one codon, as in this example, you can kind of get away with it. So this is one anticodon AGG recognizes UCC, and also allowable with wobble pairing is AGG with UCU. So I guess I maybe remember that I mentioned before G and C are both kind of swoopy letters. And on RNA, U's kind of a swoopy letter, right? Turn that C on its side, you got a U. So G and U can be friendly, make a base pair. If we take that glutamine codon again, let's draw some arrows up here. I don't know how to illustrate my wobble here. So it's possible with those amino acid codons that we established before that the same GUU anticodon, I guess I'm thinking about these now, 3' to 5', but that same anticodon could recognize CAA and CAG because of the third position wobble pairing. So the anticodon is it's a specific part of the tRNA, kind of at the business end of the tRNA. It sticks out and recognizes the mRNA. There's a whole bunch of other nucleotides that make up the tRNA. They're not responsible for recognizing mRNA. But at the other, I guess that's the other business end. The other business end of a tRNA has a location where a specific amino acid is added, and that allows the codon to match a unique amino acid. I don't know, more or less unique. Some version of unique. All right. How's that do? Making sense? Okay, so I know we have the exam coming up next Thursday. We'll have really the end of content that'll go onto the exam we'll cover by the end of next class. I was trying to get through a little more mutations today, so we'll get it started today. We'll finish it up next time. You have seen mutations in lab. So the fruit flies that you started with, those would have been discovered initially as mutants. Holy cow, I have a brown-eyed fruit fly. It's a mutant. What is a mutant? A mutant is an organism that is mutated for something that we find interesting. Mutate, the term mutate just refers to a change. And in genetics, a mutation is a change in the DNA sequence. So I guess keep that in mind. We're using kind of a narrow, specific definition. These changes in the DNA sequence don't have to make any changes to the organism in its function, its appearance, anything like that. You could just have a little different bit of DNA, and that still counts as a mutation. So in the yeast lab, when you zapped your yeast cells with ultraviolet light, a lot of times we don't see any growth effects. But there's a really good chance that we have nonetheless mutated the DNA of those yeast cells. So that's really where the field of genetics has required a lot of work, is making the connection between, okay, we changed the DNA, what did we do? Or how much DNA do you have to change to get a phenotype you can study? Something like that. So a couple different kinds of mutation. Changing one nucleotide into another, adding or deleting one or more nucleotides. These are kind of standard mutations. I don't know if I really have it on here. Yeah. Kind of skipped a lot of it. Where do mutations come from? Noah.
Aren't there like two main ways that it happens? One is just like crossing over and things like that happening when you're reproducing cells in like meiosis and mitosis. And then isn't the other through like radiation and things like that where it actually affects the structure of the DNA?
Yeah, I think you could probably say that. For those of you who aren't on the microphone, Noah offered that we have some that are result of mistakes in, I'll just say maybe cell division, kind of in the broad sense. So crossing over, DNA replication. And other ones come from more external factors, mutagens or things like that. And I think that's probably a good way to frame this. So one way to think about them is mistakes. I've kind of hinted at these before. During the process of DNA replication, we have to take a whole genome worth of nucleotides, copy those letters one for one, billions of nucleotides. A, G, G, A, C, A, T, T, A, G. There is a rate of mutations, mistakes that happen during that process. It's just inevitable. You can be the most careful note taker ever. If you're copying an entire book, you're probably going to make a mistake here and there. So just enzymes, sometimes a little molecular slip, put the wrong nucleotide in across from, so like a non-complementary nucleotide pairing. We also saw chromosome level mutations. Those are probably worth mentioning here. So crossing over. Separating. So we had this topic of chromosomal mutations, basically. So extra whole arms of chromosomes that are translocated between non-homologous chromosomes. A lot of times these big chromosomal mutations, those have big effects. So if you're crossing over a big chunk of chromosome, that affects a lot of genes at the same time. If you affect a single nucleotide or a few nucleotides in one gene, that's a lot less, tends to be a lot less serious. Don't push the button. External factors. So I guess the difference here is the healthiest organism can still have these random mistakes in their cells as their cells are copying. This is kind of inevitable. There's going to be a certain level of background mistakes. There are environmental exposures that can increase your chance of having mistakes. As Noah pointed out, they can cause chemical changes to the DNA. So ultraviolet light, for example, causes what we call cross-linking, binds a couple of nucleotides together, and then when that gets fixed, sometimes the fixing of that error causes a mutation in the DNA sequence. So a whole lot of UV light, it's well known, that causes DNA damage. And the cells can fix a certain amount of DNA damage, but too much or mistakes in the fixing, those can cause the genes to be mutated. This is why sunlight is a risk factor for skin cancer. I saw a robot that was disinfecting a hospital, just shining UV light and going all throughout the room. So you can basically kill if you have bacteria on surfaces or even viral DNA. Exposing those to UV would effectively disable them. One way to sterilize, we cannot, though, inject it in our veins, as some people have suggested. Right. So, right, if you're disinfecting, obviously, can't risk the health of the organism you're trying to disinfect. So kind of related, we have this term mutagen. This is basically a word that's built out of mutation and gen for makes it happen. Carcinogen, the carcino part of that is cancer. And there's a lot of overlap between mutagens and carcinogens. Basically, if you mutate cells enough, this can be the starting point to developing cancer. So I mentioned UV, ultraviolet light, as a risk factor for skin cancer. There are dietary and life choices that people can, so smoking, for example, is tied to lung cancer. Exposure to environmental hazards at work can cause cancer. Maybe you've seen these labels, right? Known to the state of California to cause cancer. Seen those? California knows more than we do, apparently. No, it's basically that they have a law that is much more strict on preventing chemicals that might cause cancer from getting out into the public. There's some gray area sometimes, right? So how much risk is acceptable? We could avoid ultraviolet light entirely and never leave our house. So it's kind of a relative amount of risk. And the California laws on there are basically, you can choose to do what you want with this, but we're going to tell you that it does have just a little potential to cause cancer. And cancer is a whole fun topic we could talk about some other time. But big connections to genetics with cancer, obviously. All right. So if we have mutated a gene, we're going to spend a bit of time talking about what might be the effect on the organism. So I suggested earlier, you can mutate DNA and not have any effect whatsoever. You can mutate a gene and have a big effect. What does that depend on? Here's an example. This is a gene that is implicated in cystic fibrosis. That actually is called CTFR. That's CF is cystic fibrosis. So this is a protein that's basically understood now, I guess to my knowledge, was discovered after they knew about cystic fibrosis, and this was implicated as the reason why. So cystic fibrosis is connected with having a mutated version of the CTFR. This is a transport molecule. And in the mutated version, it doesn't do its transport job, and mucus builds up on the outside of the cell. And that leads to a whole organism phenotype. So this whole disease called cystic fibrosis has its origins in one faulty protein. So it's possible if you have the gene for cystic fibrosis, you can mutate the normal version into the mutated version that can't do its function. So a little more of the extreme end of effects from mutations. What do I think about? If you think about a protein, we're kind of get still into biochemistry here. Proteins are big globby molecules in a lot of cases. And they're made up of amino acids, and they have some of those amino acids that are really important, and some of those that are maybe a little more expendable. So some of the places where amino acids might be important, if you've got amino acids that have, say, a hydrophobic quality that allows them to be located in the membrane, if that got mutated and that hydrophobic amino acid became a hydrophilic amino acid, that would affect the ability of that part of the molecule from being in the membrane. Other features, active sites, so enzymes that interact with molecules to do synthesis or break molecules down. If you mutate the active sites, that's how they recognize where they need to act. That's how they do their enzyme job. and mutating the amino acids in that location would have probably a much more drastic effect. So this is one I like, a nice diagram here. This is another just basically schematic of a protein, and it interacts with this molecule called DON. And what's shown here, these are the specific amino acids that actually themselves interact with DON, and they're abbreviated with the single amino acid abbreviation. So Y for tyrosine, V for valine, and then a number. So this is the 484th amino acid. This is the 249th amino acid. So just depending on how those amino acids fold up into this three-dimensional protein, these ones end up being more important for that active site. Another feature of proteins, phosphorylation. I guess we don't really talk about that, but this is a way that proteins can be involved in signaling. And if that signal is not able to be attached to a protein, then it can't do its job either. All right, I think I'll probably stop there. This has been a long day, and it is warm in here. Questions? Cool. All right. Then we will be back here on Thursday.